and for those of you that don't know what that's about. So Bob, please take it away.
Thank you, Sandy, for that very nice introduction. This is a talk that I've given a fair amount before. Probably many people have seen this. So if you have, just talk among yourselves, and we'll sort of keep going. This is evolution. This is actually a ductal carcinoma in situ, so it's a breast cancer. This is a duct full of tumor. The basement membrane is the sort of circle around it. What happens is the blood vessels stay on the outside of the basement membrane. And so oxygen, estrogen, glucose, all has to diffuse centrally into the duct from there. And as you go along the radius, it's of course being consumed. And what you see there in this staining is for GLUT1. So you can see that the glucose transporter is increasing its expression as you go in, as the oxygen concentration goes down. We tend to think about, cancer biologists tend to think about cancer as a genetic process, a mutation process. But actually Darwin didn't know anything about genetics. He didn't know anything about any of this. He only required a mechanism of inheritance. And so while mutations can provide the mechanism of inheritance, so can epigenetic changes, and so can a niche construction, which provides an ecological inheritance. And it's a match. What you're seeing here is the phenotype of the tumor matches the environmental selection forces. It's that logical connection of, in Darwin's case, the beak and the seed that provided him the insight about evolution by natural selection. When we look strictly at genetics, we lose that phenotype environmental connection. And so I think we often, are looking at the most difficult data source that we possibly could. It is an interesting, I think, Gedanken to think about whether Darwin, as a modern day molecular biologist sitting on the Beagle with his microarray machines, being brought finches by sailors tramping through the Galapagos Islands, grind them up, get the genetics. Could the modern day Darwin have written on the origin of species? And so as we talk about this, I want to kind of emphasize that we're not going to really talk about genetics very much. We're really just going to try to talk about the level of the phenotype and environmental selection forces, which I think this illustrates well. So I'm going to do a pop quiz. These are dandelions that are out in the field. They're about 30 centimeters high. Typically, they give rise to about 3 flowers per year. This is what you see on your lawn. This dandelion is single. It's maybe a couple of centimeters high, and it gives rise to the seed pod once a week. Okay, so you could look at the genetic differences between these, but what are the evolutionary dynamics? Maybe one of the students can tell me. It's the lawnmower, right? So this is the lawnmower. It's low because it wants to stay away from the blades of the lawnmower, because Americans at least mow their lawn typically once a week. It gives rise to a seed pad once a week. And my point is that if the genetics will tell you a whole lot of information, but will they fundamentally tell you about the selection force and the adaptive strategy? So just to kind of keep that in mind as we think about selection. So why do we need to understand cancer evolution? This is, I think it's probably obvious, but in the era of molecular oncology, this idea of individualized patient care, we talk about the molecular targets. This is a EGFR mutation, lung cancer, metastatic through the lung. It's treated with erlotinib. Any number of other drugs are now available, and got a great response. It's a triumph of modern molecular medicine, but the problem is that we keep treating and it recurs. The cancer has evolved resistance, it's progressed. So this wonderful response really ends up ultimately just delaying the inevitable. a general principle is that it appears that resistance to any therapy can evolve through a wide range of genetic and epigenetic mechanisms, and we'll talk about that a little bit. I think that resistant phenotypes are probably inevitable, but the presence of resistant, and so I am saying that because I think previously what we've tried to do is target the mechanism of resistance. I don't think that's going to be very useful. But a small population of resistant phenotypes is not significant until it proliferates sufficiently large to become a clinically apparent cancer. And that proliferation is governed by Darwinian forces. And so that's what we'll talk about. I don't think we can prevent resistance, but I think we can prevent the proliferation of resistance. And I would argue that these are highly dependent on the tumor, the patient, therapy, and are equally sort of personalized oncology issues that we should be thinking about. Cancer cells, like all living systems, are governed by evolutionary first principle, and this gives them an enormous advantage. They can devise genomic and epigenomic mechanisms of strategies using the human genome as a source of information. But evolution also imposes constraints and limitations. And I think those can be exploited to optimize therapy. And so that's kind of the theme of what goes on next. If you think about cancer as a game, it's a game between the oncologist and the cancer. The oncologist plays the game by applying the therapy. The tumor plays the game by evolving an adaptive response to the therapy. If you look at it as purely a game and just do a game theoretic analysis, the oncologist actually has two huge advantages. One is that he or she plays first. These are called Stackelberg games. In other words, the tumor cannot begin to play a move, play its adaptive strategy until a specific therapy has been applied by the oncologist. I think more importantly, the oncologist is sentient and can play dynamically. I knew you were going to make this comment, and I'm not going to get involved in that sort of thing. But can play dynamically, can understand the rules of the game, can change things, can anticipate an adaptive strategy, and then use that information to benefit the patient. In contrast, evolving populations can only adapt to the here and now. They can never anticipate the future. They can never adapt to a perturbation they have not seen before. And perhaps relevant to Jake's point is that for 5 decades, the standard dogma in oncology has been that a drug or set of drugs is applied at maximum tolerated dose continuously until progression. And what I would suggest is that therapy actually specifically gives up the two advantages that the oncologist has. Let's just kind of look at this again in an evolutionary point of view. Let's start with a group of cells, most of which are sensitive to your therapy, but 2 of them or a couple of them, small number of them, are either de novo resistant or can develop resistance sufficiently fast that they are not going to be killed. So give maximum tolerated dose. You get a great response. Looks great. But now what? Give the same dose, give them the same treatment, but you're treating resistant cells. It's pointless. And because you're not changing treatment, the cancer cells don't have to change their adaptive strategy. They just keep on doing what they're doing. Often oncologists say, well, we keep treating and the tumor's under control, but what in fact is happening is underneath all of this and out of your sight, the tumor is growing both in size and, more importantly, I think, in diversity. As it fills new niches, develops new adaptive strategies and phenotypes, and ultimately becoming sufficiently large clinically that it's visible and is clearly a recurrence or progression. Notice now that here the oncologist has ceded control of the game to the tumor. In other words, the therapy is changed when the tumor progresses. The tumor is leading the game. And so a lot of what I'll talk about now is trying to kind of reverse this, to get the oncologists to use the advantages they have. So here's one approach, and there are many approaches, and This is one that we've started with. This is called adaptive therapy or something like that. Again, take that same mix of tumor cells that are sensitive and resistant. Rather than giving the maximum tolerated dose, you give actually the minimum necessary, the minimum amount you can get away with. You explicitly leave behind cells that are sensitive to your drug. You want to maintain them. You then stop therapy. The tumor will regrow, but now you're applying no selection for resistance. And in general, the sensitive cells, because they don't have to carry the machinery of resistance, in the absence of treatment, are fitter than the resistant guys. And so they can use that advantage to essentially recapitulate at the end of this cycle the same population dynamic, same population distribution that we had in the first place, and you can just continue then to cycle the therapy through this. So here, treatment is really viewed as a forcing function, one that is applied at the correct time. It has to be done at the correct time to induce an oscillating your steady state. The first, we have done preclinical studies on this, and there are other strategies that can be used to exploit evolution. So I don't want to just say this is the only one, but we have started this in patients. We used abiraterone, which is a second-line therapy used in men with castrate-resistant prostate cancer. The protocol was built on a math model. So basically what we did was define 3 subpopulations based on their interactions with testosterone. The T plus cells require exogenous testosterone. Those are typically the dominant cells at the time of presentation of a prostate cancer. The TP cells are the ones that make testosterone. Those are the ones that are sensitive to abiraterone. Notice the potential to produce a common good. In this case, the testosterone they make spills into the environment. And so it can promote the proliferation of the T plus cells, which then are cheaters in this. And that's the official evolution term. And then the T cells, the T minus cells are the bad guys. Those are the ones that will proliferate independent of the testosterone concentration. So we developed a mathematical model, did a matrix payoff, and then we proceeded. Dr. Jinsung Zhang was the brave oncologist that started this. The strategy was very simple. We basically gave abiraterone. When the PSA would cross the 50% line, it was 50% lower than pretreatment values. You stop therapy, you wait for the tumor to come back, and then you resume the treatment. The simulations predicted that we would control the tumor from 2 to 20 cycles, and importantly, was incorrect. So we'll talk about that in a minute. These are the results as of actually January. I haven't really looked at them since then. So what we have achieved, we compared to a group of patients who had a 50% or more, a greater than 50% decline in PSA at the administration of abiraterone, but then went on to have standard of care, maximum tolerated dose until progression. The median time to progression was increased by 19 months. Overall survival was increased by more than 2 years. Because the patients were on off, they were receiving abiraterone only about half the time. And so were being treated actually only about half the time. Abiraterone is fairly expensive. And so this was analyzed by a group of economists who said that there was a reduction in patient costs by about $70,000 per patient per year. And then there was a surprise, and that is this, and you can see this curve tails off. There were 4 patients that remained cycling even 7 years out. We did not predict that. As I said, the model predicted that there would be a conclusion to every patient. And so that was one of the reasons that we started to look more carefully at our data. Now, I just want to do a plug here, and I know I'm preaching to the choir here, but the current clinical practice is focused on these double-blind randomized trials that often involve thousands of patients that can cost up to $1 billion. And at the end of that trial, what you get is that you've looked at A and B, and you can get a statement that A is better than B under these conditions with this level of statistical certainty. What was never asked or answered is why. why is A better than B? Could we have done A better than we did? Could B have been better if we had done different things? And probably most importantly, what do you do next? You know, if A is going to be better than B, then if we have C, how do we add C to A in a way that makes optimal response from patients? So what we could do here is then use the mathematical models that were used to design the trial to also analyze the trial. Okay? And the way we did this, and I'm just going to gloss through the details, this has been published, is that we take the longitudinal data from the trial patients and we upgrade, or we check the parameter estimates. And importantly, one of the parameter estimates was wrong. And that was the ratio of the fitness of the sensitive population to the resistant population in the absence of treatment. We had thought that it would be, the models showed that it was two or three. And what we found is that the data suggested it was more like 6 or 7. This turns out to have clinical significance, oddly enough. Let me see if I can walk you through this. So this is assuming that this ratio is 2 or 3. Now look at what happens in each cycle, and as you treat, the sensitive population declines, and because of that, the resistant population will increase. What the model predicted is that when you stop therapy, you will see an increase in the sensitive population, and the resistant population will plateau. So what you get is an up plateau, up plateau, up plateau, but a consistently increasing number of resistant cells. And then that is the reason that we had this upper limit of a cap on how many cycles that we thought that we could control the patients. If it's actually 6 or 7, what happens is that instead of this plateau, when the sensitive population goes up, the resistant population will go down. And you can see that on the right. You can actually, with a steady cycle, you can actually drive the resistant population somewhere close to extinction. So now what you can do is, once you have the model, you've updated the parameters, you can now, we apply the model to every patient in the trial. And the model tells us what happened. This is one of the patients who's now actually 7 years into treatment. And what the model showed us was that early on, we had these cycles that worked really well, where we actually hit the right kind of cycling with the sensitive cells, driving the resistant population somewhere close to extinction. And what the model suggested is that it was not increasing throughout this treatment. So 2 consecutive ideal cycles. And there's a couple of things about this. Notice that the troughs of the actual measured, which is in the top row, do not change. What we found was that as the resistant population increases, those troughs tend to increase. So we're seeing this flat, steady trough. And we've also done serum markers for resistant, for DNA for the resistant genes, and have found those to be negative. So this is a person that just keeps getting treated. Now, why did we fail? Which I think is probably the more important question, and I think a question that we really need to be answering more and more of during therapy, when we evaluate our trials. And the reason is we made a mistake. We made an error in the protocol. We agreed that we would stop when the PSA dropped to 50% below pretreatment value, but we required that it be confirmed by radiographic studies. Now, we can do PSA every month, but we can only do radiographic studies every 3 or 4 months. So what was happening was that, you can see here, this patient we should have stopped therapy here. But in fact, it went down to 0. And before we stopped therapy, and what happens is that we're reducing the sensitive population. You remember, this is unlike what's intuitive, where you want to kill as many cancer cells as possible. Here, we want to keep those guys around. And the more we kill, the worse we do. And what we found was that very consistently, essentially all the patients that we saw progress is that we had these episodes of these periods in which our timing was incorrect and we overtreated the patients. I'm going to change topics now, and I'm going to talk about the evolutionary dynamics of potential cancer cure. I think that since the time of Ehrlich, who was a brilliant chemist in the early 20th century, who proposed the idea of magic bullets, we have been looking for them. The magic bullets are drugs that can selectively kill cancer cells, but not normal cells. He proposed that these would be something that could be found, and certainly the antibiotic revolution of the mid-20th century it reinforced that idea that we should be able to find them. To be honest, my hypothesis before I started this was that the cure is not possible. The size, the diversity, the spatial dispersion of a typical metastatic cancer are insurmountable barriers to any attempted cure. But I was kind of ordered by Dr. Damon Reed here, who is a good friend and one of our many collaborators, who treats kids with metastatic sarcoma. These are teenagers who, once the disease is metastatic, almost certainly die of their disease. And his comment was that adding several years of life to a teenager is nice using adaptive therapy, but we're about cure, and you really got to do better than that. And so this group of us in here have been looking at a variety of different approaches to can we use evolutionary dynamics for cure. So arguably, cancer cure is essentially an extinction. Several people have written about that. I know Carlo has an article on it, and I completely agree. I think that, at least for me, and I think for most people, the popular imagination always goes to the extinction of the dinosaurs when we talk about extinctions. The application of a massive amount of evolutionary force, single event, single cause, wipe them out. And arguably, the way we treat cancers now with maximum tolerated dose, maximum dose density, has some elements of this kind of extreme application of evolutionary force. But the problem is that we tend to forget that the KT impact also eradicated about 60% of the land animal species at the same time. So brute force as an extinction strategy is really good, but it has a lot of indiscriminate effects. And if you are trying to treat cancers, the indiscriminate effects are going to be related to normal cells. And I think it's probably fairly obvious that we're always being limited by that, by the toxicity. So the question is there an alternative? And so really the questions are, is the dinosaur extinction the only kinds of extinction? And the answer is no. We live in the anthropogenic era, the Anthropocene era, which means that it's an era in which our species is killing off other species. And that's a tragic outcome. But it has a silver lining because it's allowed us to watch species extinctions in real time and to study them in detail. And from that has come a very different view of extinctions. And I'm going to give you an example of an intentional Anthropocene extinction of the Galapagos goat. The pirates and sailors in the 19th and 18th century would stop by the Galapagos for layovers, and they left goats there because they wanted to have food when they came back. These goats became feral and they began to proliferate. And so by the 1980s and 1990s, the population of the Galapagos goat was in the range of 500,000 individuals. And it was basically crowding out the native species. It was eating a lot of the native shrubbery. And it was decided by the international community that it should be made extinct. And this was called Project Isabella. How do they do it? Well, they took New Zealand sharpshooters, gave them automatic weapons, put them in helicopters, and they went around the islands basically gunning down the herds. And this is the gruesome outcome. Populations, of course, plummeted. And then they stopped plummeting. Why? Because some of the goats developed the ability to hear the helicopters or the trucks coming over them, and they would run into the woods and hide. And these small populations were therefore resistant to this mechanism of eradication, and these populations began to grow. So you could give, I mean, you could put 100 helicopters flying around with New Zealand sharpshooters all the time, but these guys knew how to, they were resistant. So continuing this was futile. And what did they do? They developed a strategy called the Judas goat. This is a female goat who was neutered. They put a radio collar on her, poured hormones all over her fur, sent her out, and she joined these small herds. And using the radio tracking, they could locate the herds and kill them. And so one by one at a time, they eradicated these resistant populations, and the extinction event took place. Now look at this study out, because this is actually typical Anthropocene extinction. There's a first strike in which there is one or a series of perturbations that take the initial large population and greatly reduce it. They typically produce resistant populations, and those are typically fragmented spatially. The final nail in the coffin of a species is typically a different nail than was then applied in the first strike, often a sequence of nails. We'll talk about that in a minute. But the slide to extinction is then by a different second strike agent. Notice the difference. The first strike was very effective, but failed as a second strike agent. The second strike agent would never have been effective as a first strike agent because the population was way too large. It's this combination. And so Anthropocene extinctions typically go through this cycle. And what's important is that they're caused by a sequence of perturbations, none of which by itself could cause extinction. It's the combination, it's the strategic sequence. Is this relevant to cancer? Well, this is an example of, this is a breast cancer pre and post neoadjuvant therapy. On the left is the pre-treatment. You could see a continuous population of cancers that are contiguous and heterogeneous. On the right, what you see is the sample, the surviving population are small islands of cells scattered between in this area of necrosis or fibrosis, fragmented, very similar, I think, to the Galapagos goats kind of surviving populations. So one of the things that I think we have not done, at least as best I know, is to really understand, why do cancer cells survive in vitro? So we can do it in culture, but let's do it in people. And so this is now taking this pre and post neoadjuvant therapy, breast cancer, and we're looking at the survivors only. And you can see again, you have these islands of cells, but these are now, notice that they're relatively differentiated. They're forming pseudoducts, something that we had not expected and something that was not seen in the pre-treatment tumor population. And we're starting to look at their properties. And one of the things we've learned is that the proliferation rate is zero, as far as we can tell. These guys are not proliferating, nor are they dying, but what they are is metabolically very unstable. They're very stressed, high levels of expression of VEGF1, HIF1, CA9. So we're learning some properties about them that we've seen seem pretty consistent, but these are potential things that we can exploit in terms of second line therapy. The other thing we've learned, and this is work with Asma Kanawa, who is, it was just this earlier this year, is that when we look at cells that are surviving, we can see some that begin that have a pre-existing proliferation, pre-existing resistant mutation, and they begin proliferating very early. But there's also groups that actually sit there quietly, apparently quiescent, for months, and then begin to proliferate. And that was the group they were particularly interested in. And what we found is that those cells are identical genetically to the parental cell population. There are no genetic mutations. There's no resistance mutation. Everything that they've done has been involved with epigenetic modifications. You can see on the far right the difference between the methylation patterns in the two groups. And one of the things that we've learned and most is you can kill these guys with hypomethylating agents. So they have a weakness which you can potentially exploit. So the idea then is to begin to really categorize the mechanisms of resistance and begin to define methods of exploitation. Asma, by the way, is joining the University of Indiana as a faculty member and is recruiting some postdocs if anyone is interested. So if we now review this, and let's say that we have in the top row here the U shape of a continuous therapy. You get a very good response. You get this sort of stability for a while when the tumor is not really visible, and then it begins to grow. If you give therapy when it begins to grow, it's too late. It will just continue to grow. If you add a second treatment at the time of the first treatment, you're applying both therapies at the maximum size of the population. Therefore, the probability of finding populations that are resistant to both is at its highest. However, that does, as the models show, tend to increase the time to progression, but does not increase the probability of cure. On the other hand, when do you give therapy? When does the second strike most effective? It's at the point of maximum response or near that. And you can see on the right that if you now apply your second therapy at this vulnerable point, when the population is small and damaged, that you actually have at least the potential for eradicating the population. But this turns out to be very time dependent. So we're going to give a second therapy. This is the beginning. We're going to give a therapy at the time of this little point. So first we're going to give it right at the beginning, and then we're going to give it during this sort of trough, and then we're going to give it late. So you can sort of watch how this. Okay, so you see that you could actually get an eradication of the population, but only through this very brief time. So this is highly dependent on. I'm not going to do that again, but it's highly dependent on the. Okay, there's effective. So too early, too late. You missed the boat. So the timing has to be very exquisite. This now gets us into the real problem, which is how do we understand internal evolution during therapy, and we just don't have good biomarkers. But this is the idea of an extinction therapy, that you give a first strike agent, you get a very good effect, but you select for resistance. Now you give a sequence of additional perturbations that exploit the adaptive strategies that we've seen and hopefully will continue to see in a way that just keeps reducing the population. This sequence of events is actually well described in the evolution. It's called the extinction vortex. The small populations are vulnerable to extinction because of statistical changes. In other words, a small death or a change in death rate or birth rate in a small population can drive it to extinction, whereas you would never see that in a large population. The other thing is that they lack allele effects. And so the ability to resist perturbations lacks the herd effect that you can get as a larger cancer population. Each time you reduce the population a little bit more, those vulnerabilities get greater. And so when you do this extinction, this sequence of perturbations, they do not have to be severe. They don't have to be prolonged. All you have to do is just keep nudging it down this extinction vortex. The smaller the population is, the more vulnerable it is to subsequent perturbations. And so the idea then is to harness the extinction vortex for curative intent. And our first clinical trial on this is in metastatic prostate cancer and has accrued 16 patients with a goal of 32. And at 20 patients, we will stop and do an interim analysis. So that will be probably done pretty soon. So these are the lessons. So let's just go through this. This is a typical prostate cancer patient. You give androgen deprivation therapy. The PSA goes down, stays down for a long time, comes back up. You then give second therapy here. What we propose is you give second therapy there. And at the peak of response, you basically kick it when it's down. You hit it again and again and again with, again, the right sequence of events. If this sounds familiar, it is actually. It's actually the empirically derived treatment for ALL, for pediatric ALL. Again, an induction period. What they learned was that at the end of an induction period, you often couldn't see tumor in the bone marrow or the blood, but what they learned historically was the tumor always came back. And so what they decided to do was, rather than wait for it to come back, is to treat it early, immediately following this induction period with the consolidation. So this is a first strike, second strike, third strike kind of approach that is exactly the kind of thing that we think could be applied to adults. I should also point out that this exploitation of small population dynamics is something that we should be thinking about in terms of adjuvant, neoadjuvant therapy, where the explicit goal is to eradicate a small population. We should also be thinking about prevention, where again, at some point, you're dealing with a small population that should be vulnerable to those extinction events. And so with that, I will thank the many people who are involved, many of whom are in this room. I will just throw in a plug for the Moffitt Evolutionary Tumor Board, which is, I think, quite unique. We actually have a tumor board run by oncologists, but attended by mathematicians and evolutionary biologists, where we explicitly talk about the evolutionary dynamics and how they can be exploited for the benefit of the patient. So with that, I thank you and happy to answer questions.
That's a great talk. I'm curious about a few things. One, it seems that the clinical problem is really about adaptability, resistance being one form of adaptability, and that cancers actually can make the first play in this. At the time of diagnosis, cancers can have subclonal architecture with different clones that are more or less adaptive. They can have epigenetics that make them more or less adaptive. They can have genes that are poised to be expressed through known mechanisms. So in that, cancer cure rates are about 70% now, which means that the things that we've been doing historically actually are curative in most cases. It's not that repeated treatments result in the inevitable emergence of resistant cells. That's not the case. And so it would seem to me that it's really about the clinical problem is addressed by assessing, if you can, at the time of diagnosis, what the adaptability of an individual patient's tumor is. So for instance, we do drug screening with biopsy isolates from brain tumors, get clinically actionable treatment recommendations. But moreover than that, you could just set up different culture conditions, right? You could have plastic, you could have slice culture, you could have organoid culture, you could have, floating culture. Those cells, we know that most cells can adapt to each of those things equally rapidly, like when you take a patient specimen and put it into culture. It would seem that those patients that are gonna need some kind of adaptive therapy rather than standard approaches could be identifiable at the time of diagnosis by whether or not their cancers are adaptive, either to different culture conditions or different drugs. I'm wondering what your thoughts are.
There's a lot of parts to that.
But there's one central part. Adaptation is the problem. If cancer cells can adapt to the selective pressure of treatment, they recur. Right, that's the problem. It's the recurrence that kills patients. If you could identify, let's just call it an adaptability quotient.
So what would be a metric for that?
The ability, so let's say you had 10 different culture conditions that ranged from, plastic, coated plastic, fixed monolayer of cells, co-cultures, organoids, slice cultures, stem cell culture. So let's say you had the whatever number that is.
Possible. But also remember now, they're not being faced with the selection pressures that they are in the body.
Those are the selection pressures, I'm going to say. Those are the models. Culturing on a slice, culturing as an organoid, you're recapitulating the native environment
some
to
of
the
it,
best
but
of
not all
our
of it.
ability.
you don't have the immune system, you don't have androgenics, you don't have other host response mechanisms of this, and how
You don't have everything.
we don't know
It's
what
true.
they play. And so, yes, I mean, I completely agree that once we understand the metrics of that whatever confers adaptability, and whether, it's possible that what you've suggested would be a metric of adaptability that was usable. I'm just not really sure. One of the things I've learned is that cells that adapt to a culture dish are really different from cells.
Exactly.
I mean, you could see they diverge immediately and significantly. So let me just say that one of and the other thing is what is adaptability driven by? Is it driven by mutations, which I actually don't think, or is it driven by epigenetic mechanisms, the ability to access the human genome?
I think it's both. Subclonal architecture, definitely identifies pre-treatment clonal diversion through mutation, and then epigenetics on top of that. So I think it is both. And that's why I think assessing that, the adaptability, and it could be done like in your clinical trial. You could be setting up these cultures, looking at adaptability at the same time that you're measuring patient response to adaptive treatment, right?
Sure.
Does it correlate the ability? I mean, that's what needs to be done, it seems to me.
Yeah.
On a practical level.
Let me just, I agree. Our top priority has been to understand the mechanisms of resistance what's the distribution of mechanisms of resistance, to just get some sense of what are potential second strike agents that we could exploit these things. Now I suspect once we have that, we will be able to go to the beginnings and say, these are the things that they can evolve into. Are we seeing that ability there? But I don't know. This is like
we could talk more later.
I said this is like washing up on North America in the 1400s. I mean, there's like a whole continent of things to look at, and it's hard to sort out.
Yeah,
and it's not necessarily easy to get funded. So combinations.
We could talk. I don't want to take up more time.
I did want to mention how but it was a paper
I think he's going to present this afternoon. Is he going to talk a bit about that model? Yeah, because it was a highly plastic, very mixed phenotype model. You could jump from resistance to one drug to the other to being double resistance with a mutation rate. So there's some interesting results that come out of that.